All right, so for this particular lecture, we're going to be taking a look at your textbook, Chapter 5, and we're going to focus in on the plasma membrane. Now, we've already talked about the plasma membrane in some contexts because you need to be able to talk about membrane-bound organelles. This time we're going to look at the plasma membrane, the surrounding outside cellular membrane, as its own organelle and some of its functions. So here's our first list of terms to know from this chapter. Some of these things are going to overlap with our next lecture as well, so you want to make sure you keep track of this one before you move on to the second lecture in this series. So we're going to talk once again about our plasma membrane. And remember, plasma membrane is one of our 4 things that we find in all cells, right? Plasma membrane, DNA, cytoplasm, and ribosomes should be found across the board in all of our cells. There are differences between plasma membranes in prokaryotic cells and eukaryotic cells, but those are on a molecular scale that we're just not going to get to for this course. If you get into microbiology or you take a separate cell biology course, that's going to be of more interest to you. Right now, we're going to look at the things that are universal to plasma membranes, things that we find generally, whether in a prokaryote or a eukaryote. So do remember that our main function is to separate the inside of the cell from the outside of the cell, right? And in order to do that, in order to make a distinction between what's on the inside and what's on the outside, you need to regulate. You need to decide what's allowed into the cell, what's allowed out of the cell, okay? And so we see there's a series of structures involved in plasma membranes to allow that to happen. First off, we have our phospholipid bilayer. So you might want to take a look back at Chapter 3 to think a bit more about phospholipids if you'd like. But remember that these are amphipathic molecules, which means that they've got a hydrophilic end and a hydrophobic end. And the hydrophilic end is going to be found facing the outside world, and another hydrophilic end is going to be found facing the inside of the cell. And then our hydrophobic tails, right, of these molecules are going to be found facing inside, making our phospholipid bilayer. But within the phospholipid bilayer, there are other molecules as well. Okay, so I'm going to show you a picture of what that looks like. You'll notice that our list here includes carbohydrates, it includes 2 different types of proteins, and it includes cholesterol, which is a different type of lipid. Okay, and these all have their own unique purposes. We're going to briefly talk about the carbohydrate chains and some of our proteins in this lecture. We're going to get it deeper into the proteins in our next lecture. Cholesterol, as a lipid, right, is going to help to adjust that lipid bilayer. So here's a picture of the entire phospholipid bilayer, and it looks fairly complex, but it's actually not that bad. If you take a look, what we see is those lovely phospholipids. Remember these guys here with their hydrophilic heads and their hydrophobic tails. The hydrophilic section is going to face the watery parts of the world, so out in the environment, in most cellular cases, right, is going to be watery. And then inside the cell itself, right, our cytoplasm is aqueous. It is water-based. So it makes sense that you're going to have your hydrophilic sections here. Your hydrophobic tails are going to build the bulk of our membrane, right? These are going to be our part that's going to separate things, right? So our hydrophilic tails face each other. We have one layer up top facing the outside world. We have one layer down below facing the inside of the cell. So this is a bilayer, 2 layers right here. Okay, so a couple other things to point out here. If you take a look at this picture, it almost looks like you've basically got a wall, right? Almost like a brick wall, except it's little phospholipids, right? All lined up in little rows and columns here. And that's because you're looking at a diagram and not at a living sample. If you were to look at this actual thing all put together, all of these lovely little phospholipids would be moving around each other, almost as if they're in a crowd of people. Right, so it's a barrier, right? It's a barrier to try and get through a crowd of people. But the individual members of that barrier move around. Okay, so this little guy here could move 3 phospholipids over to the right or to the left. You even see in some cases you can flip them from one side to another, though that takes energy to do. So that's our phospholipid bilayer setup, and you'll notice that the entire membrane is not just phospholipids. It also contains proteins, some of which go all the way across both sides of the bilayer. Okay, some of which just sort of sit on one side or the other. Some of them sit adjacent and never actually cross into the bilayer at all. We have proteins that are attached to sugars, which are called glycoproteins. Okay, we also have sugars that are attached to lipids, which we're not going to get to here. Here's one here, a glycolipid right here. And we have our cholesterols. Remember that cholesterols are steroids, so we have that 4 chains of carbon, right, going, or 4 loops of carbon going in right here. So that's our total sort of setup here. We've got a lot going on. Lipid bilayer, We've got our proteins, we've got some carbohydrates sticking out on the outside world end of our membrane, okay, and we've got some cholesterols in here to help to stabilize this whole shape. Keeping in mind that phospholipids can shift around and change their relative location. What cholesterols do is they actually prevent the cell from getting a little too wiggly when it's warm and a little too rigid when it's cold, okay, by being lipid in the midst of phospholipid. Here's a little summary of what I was just talking about, right? We have our hydrophilic heads and hydrophobic tails of our phospholipids themselves. We have our bilayer, right? The fact that we've got those two layers, one above and one beneath, okay, with the head of each phospholipid facing away toward the water and the tails facing each other. And like I was describing earlier, remember that those molecules are not sitting there static. They're not stationary. They're flowing around each other, which is what we sometimes call the fluid mosaic model, right? So fluid because they shift, mosaic like the art, right? So a bunch of small pieces that are put together to build something larger, okay? So like tiles or something like that. Only in this case, the mosaic is our phospholipids and our proteins and our carbohydrate chains and all that good stuff. So that's our general structure that we'll be looking at. We'll talk a bit more about how the phospholipids themselves work as a barrier in the next lecture. I'm going to show you the different types of proteins that we find in here because those turn out to be super important, and we've actually talked about some of them already. So this particular picture is found on page 85 in your textbook. It's figure 5.3. All right, we've actually already talked about one of these guys here, the junction proteins, the things that allow cells to molecularly attach to their neighbors, okay? These guys we've already talked about. So, like I said, see Eukaryotic Structures Part 2. You can go back, take a look, and read up on these different types of junction proteins. These other types of proteins, though, that we find in cellular membranes have different types of jobs, okay? Channel proteins and carrier proteins are going to allow stuff to go through the plasma membrane, okay? So if you think about it, right, phospholipids aren't going to let just anything sneak through. They are amphipathic, right? Hydrophilic heads, hydrophobic tails. In order for something to pass through here, it needs to be able to follow the rules of the hydrophobic tails in particular, which means if your cell wants to get something through its membrane that is hydrophilic or ionic, then it's going to have to use some other doorway. Channel proteins, those are doorways. Typically we use the term channel protein when there's no barrier there, or it can simply be open or closed. There's no sort of specific lock mechanism, if you will. We use the term carrier proteins for molecules that actually alter their shape to pull or push, if you will, an atom or molecule across that membrane. All right, so these are a little bit different because they're not simply an open door like this is that's going to allow stuff through. These are all based on the shape of the protein. And remember, a protein's three-dimensional shape always tells you about its function. We talked about that in chapter 3 as well. So carrier proteins' shapes, okay, are going to be really important for us as we start thinking about them. We have cell recognition proteins, which we're going to get deeper into in just a minute. Cell recognition proteins often have things like carbohydrates on their surface, okay, though not all of them are. Some have protein. Some are cell recognition molecules that might have carbohydrate with a lipid instead. The idea here is to serve as identity for your cell. Okay, so this is going to be really important in multicellular organisms such as ourselves to let that system, if you will, to let things like the immune system, for example, know that this cell actually belongs here. And an interesting twist of these cell recognition proteins is that if you don't carry ID, usually the immune system doesn't mind. It's if you have the wrong ID that causes the problem. We also have receptor proteins, which we'll also get into in just a minute. These are shaped to receive particular messages, usually in the form of hormones or neurotransmitters. Okay, they land on the hormone or neurotransmitter lands on our protein, and that causes a change inside the protein itself that's going to send a message deeper into the cell. Enzymatic proteins work like enzymes. So these guys are basically enzymes that are embedded into the cell membrane to change things as they approach them. All right, so we're not going to get too deep into enzyme proteins right now. We'll talk about them more when we talk about enzymes generally. But for this particular lecture, we're going to concern ourselves with cell recognition proteins and receptor proteins. We'll start with our receptor proteins. So receptor proteins, once again, are basically like a little lock and key is the traditional way of talking about it. They are shaped to match a molecule's shape. Okay, so they're shaped to match a particular neurotransmitter, which is something that's released by your nervous system to communicate with the next door neighbors. Okay, so if this was a neurotransmitter in our image here, it would have been released from a cell that's like right there, like just off the screen. It wouldn't be very far away. You can also do this sort of receptor protein type of thing with a hormone. That's actually what this picture is showing here. This is giving you an example of a hormone, okay? Hormones are also molecules that are sent out to signal neighbors, okay? But neighbors over longer distances. So it wouldn't be a cell right off the screen. It would be a cell in another part of the body that's creating this molecule, moving it out into the bloodstream typically to be taken up by a receiving cell. All right, or the receiving cell's receptor. Now, this brings us to a really important idea in proteins in general, and that is the idea of the conformational change. Okay, so conformational, notice that ORM. Okay, that means that this changes, right, in its shape. If something conforms to something, right, it matches its shape. It sort of molds itself to something. Okay. Your receptor proteins will change their shape temporarily to accommodate the receptor molecule. Okay, so whatever the signaling molecule. So whatever that signaling molecule is, whether it's a neurotransmitter or a hormone, should match pretty well with the receptor to begin with. But then the receptor, once it receives that signal, is going to temporarily change its shape in order to cause chemical reactions on its other side, inside the cell. All right, so our receptor's protein, in other words, temporarily changes its tertiary structure, its three-dimensional structure. That's going to cause a cascade of things to happen inside the cell. Hormones can do these three different things. You'll get deeper into that in A&P2 if that's where you're headed next. All right, and we do see hormones in plants as well as in animals. So if you're interested in plants, don't think that this is something you don't need to worry about, because this totally happens here too. All right. But the general gist of a receptor protein, right, is that it's sitting there awaiting the signaling molecule. It's awaiting that sign to say, okay, change something on the inside. We also have cellular recognition molecules, and some of these are protein-based, in which case they are called glycoproteins. Some of them are lipid-based, in which case they're called glycolipids. Now you'll notice the prefix in both of these is glyco. This is not telling you that it's a recognition molecule. This is telling you that it's a carbohydrate, okay? In both cases, these are proteins linked to carbs or lipids-based, linked to carbs. All right, and these exist in order to face the outside world, the outside of the cell, okay, to let the neighboring cells know its identity. So we see this in a couple different ways. You might have heard of blood typing before, right? The ABO blood group, so being type A or type B or AB or O, okay? This is actually what's being measured is glycolipids, right? So these sugars attached to lipids on the outside of a cell. Your ABO blood group is talking in particular about molecules found on your red blood cells. And basically what happens is if that glycolipid doesn't match the expectation of the immune system, okay, it will be recognized as an enemy. All right. Now, when your immune system recognizes something as an enemy in this way, we refer to it as an antigen, and the immune system then builds up molecules to try and attack it, and those molecules are called antibodies. Okay, so the antibodies attack the antigens. All right. When you get a vaccination, you're actually teaching your body to build antibodies by giving it a mimic of the antigen, not the real thing, like a fake version, a deactivated version. That way you have the troops ready when the real thing comes by. Glycoproteins, same idea, can wind up being antigens to the immune system. Okay. Once again, the thing that the immune system is looking for is matching. If you have a glycoprotein that doesn't match what the immune system is expecting, that cell is going to be attacked. In the case of these glycoproteins, okay, and the glycolipids for that matter, if you don't have one of these, if it's just lacking altogether, that cell is going to be ignored by the immune system. Okay, so lack of identification is not a problem. It is the wrong identification that's the problem here. And really what your immune system is looking for is actual enemies, things like bacteria or other pathogens that could potentially cause problems. We also can use these as ways to try and tag cells that might be starting to malfunction a little bit, okay? So not necessarily glycoproteins and glycolipids in these particular examples I've given you, but your immune system will look for malfunctioning cells of your own type as well to go after. So that's going to be our list for this particular section of talking about cell membranes, right? We'll look at the other types of proteins and some interactions in the next one as well.